thank you very much for the invitation. This is really nice to be here. The talks today were stunning. I should say that as the director of an formerly funded NHGRI large-scale sequencing center, I'm a big fan of this program and programs like this, because you really cannot stress enough the importance of pure technology development and then coming together as a group to share your ideas and your progress. Long ago, we did a lot of that during the NHGRI Sequencing Center days, but that is no more. So I'm glad to see programs like this succeed and to carry on. It's amazing. So I was invited here today to give a little historical perspective and a glimpse into the future. The timing is funny. So last week, I gave 2 talks that had me particularly sort of immersed in history. One of them was at a symposium at NYU, celebrating Aravinda Chakravarty's 70th birthday. So he was my PhD advisor, and he gathered together all of his students and trainees of various types, and we met to celebrate him and gave talks, and so that had me thinking a lot. And the other was the 20-year birthday of the Broad Institute. So just 20 years ago, we were founded, and I have been there ever since, as Mark pointed out. So coming out of those, I thought I'd pass along a few little tidbits. Mary Claire King gave a wonderful lecture at NYU last week for that 70th birthday symposium. And she reminded us that it was less than 70 years that it was recognized that we have 46 chromosomes. Previously, it was thought somewhere between 45 and 48. Throw a dart on the dartboard, but only 70 years to knowing how many chromosomes we have. And just 20 years since that first human genome. Karen told you a lot today about how that has been considerably made more rich and detailed in terms of variation, but only 20 years that we had that first genome, and now millions of genomes have been sequenced, and we'll talk more about that. And how about 10 years ago? So I looked back, and in 2014, our center at the Broad Institute generated 1,000 terabases of sequence data. So that's 1,000 times 10 to the 12 bases of data was generated. Sounds like a lot, right? But what about now? So in 2024, our center, with about 1/3 of the number of machines, actually, believe it or not, can generate 38,000 terabases of sequence data. So basically, we do in 10 days what took us a year to do in 2010, not that long ago. Just to give you, a sense of how rapidly things are really advancing in sequencing. To speak to scale, I guess, and overall, so the kind of data and what's been generated and how steep the slope of those curves are. So here I'm just plotting again for the center I direct, how many petabases of data have been generated, so over 100. We've recently marked producing over 1 million human exomes, almost 700,000 complete human genomes, and data of other type. And a lot of this data really underlies many of these big sort of resource projects you all know about. Mark pointed out TopMed, GTEx, all of us, TCGA, and so on. So a lot of that data is directly as part of those programs. But what I'm going to talk about today are a couple things which might seem disjointed. I hope not. I've not given this exact talk before, and I will end on time and try to leave a little bit of time for some questions. So I want to talk about human genome. It's not entirely a tech talk, but it is technology development that has led us to be able to so routinely now sequence the human genome. So I want to talk a little bit about that and how we might use it. I'd like to talk about how, I think a lot, and others, and all over the place, like we think about how are we going to move the needle now towards more sort of routine clinical use of the human genome and of genomics, and then perhaps more equitable benefit of that information across different populations. And finally, it's a technology development seminar. So I'll talk a little bit about some new technologies and some things that I think are particularly exciting as we think about not just human genomes, but also using genomic tools to really try to get to function, right, of some of these genetic associations that we're finding. Okay, so genomes. Genomes for everyone. Isn't it time? So I think right now I could get you a clinical, so an interpreted clinical genome for $500. It's pretty good, right? $500 for a genome. The genome is the best test you can perform for a genetic, any genetic screening. It's backwards compatible, if you will. So instead of doing panels or instead of guessing or trying over and over again, let's just sequence the genome. You have it, doesn't change really, for your whole life. That would cost, averaged over an entire human life, about $6 for, every year you're alive on average. That equals about 0.19% of the healthcare costs of a US person. So pretty affordable, right? And you can get, I think, a lot out of it. But a bunch of people in the room agree with me on that point. So why aren't we doing more of this right now? What's holding us back? The biggest things, who's paying? So this is always the biggest question, right? In the US, insurers are not incredibly motivated to pay for this test. They don't really see, I think, widely the ROI. And we know that for most people don't stay with an insurance plan for more than a couple of years. So who's going to invest early when that life is going to move to a different plan, right? So this is a hurdle for us to get over. Who's ordering the test and why are they ordering the test? I think it's fair to say physicians are generally not motivated to be the ones to take the leap forward. So who's going to really push them to do that? Consumer, medical societies? I don't know yet. How are the results getting delivered? So there's not a real clear path right now to excellent, I would say, decision support system integration with the EHRs. There's many different types. I came to realize a lot of these points during COVID. I'll get to that in 2 slides, but these are the things I've been reflecting on as some of the things that are holding us back. But I think we're getting there. I think now's the time. I think we're primed. So if you think about, sort of the cancer sequencing world compared to the germline sequencing world. So in cancer genomics around 2009 is when we were really taking on the TCGA, right? We were 20 different tumor types, thousands of different tumors. We're performing an atlas, this kind of study of genomes, exomes, methylation, RNA sequencing, and so on. And a lot of that data that was an understanding of different tumor types that was taken from TCGA has what then led to companies like Foundation Medicine, Guardant, Caris, Tempus, and so on, now having those panels that are provided in routine, diagnostic care for cancer patients. Here we are in 2024 with the UK Biobank, with the All of Us research program. So is now the time, right? Are we finally getting to the point where these programs are going to really spur and motivate that use in clinical care? I hope so. And I would say that it's something that, we at the Broad are extremely interested in seeing happen, and something I spend a lot of time thinking about. I will tell you that the COVID experience that Mark alluded to really did impact me and made me think about how we might make some changes as a nonprofit research institute, not your typical diagnostic company, looking to turn a profit margin. Because what we did in COVID was kind of just that. When the pandemic started in March of 2020, the local hospitals in Boston said, can you help us? And at first I thought, there's no way we can help you. I mean, sure, we have a CLIA lab that we had developed for sequencing, but we had never done a single viral diagnostic assay in our lives, right, in March 2020. But they were needed sorely. And so we stepped back and looked around and said, well, maybe we can stand up a system that could do a couple hundred a day. So a couple hundred a day turned to 200,000 a day. And we lived in that world. I'll show you in the next slide for a really rocky few years. But anyways, we did it because it was for the public good. We priced that test with no profit motive. So we sold the test for somewhere between $8 and $25 when Quest and LabCorp were selling it for $150 to $250 per test. And most importantly, we created access where there would have been very little or none. And that did sort of enable some of the schools, right, who wanted to do this testing to get back to school, to have routine monitoring. And so it did make a difference. And so it's sort of some of these principles that I keep thinking about when it comes to our genome efforts. So just to show you what that looked like very quickly, a little sidelight. Believe me, I have a 45-minute speech I do on these 3 years that I've done about 55 times, but I'm just showing you 2 slides from it. So this is what it looked like, right? So we started in the early days, just sort of like skirting along, doing that sort of couple hundred to 1,000. Then we made a big jump forward because we kind of built a whole system that got to this level of doing hundreds of thousands of tests, or capable of 100,000, most days well above 100,000 PCR tests a day. And it was rocky. I mean, you all know that for a time, it seemed like we were clear of things in 2021, then all of a sudden the Delta variant, and we had to scale it all back right up. So there was tremendous chaos. We hired 500 people in these couple of years and trained them. So quite something. Anyways, that's over now. Our last test was last June, and we're back to genomics. But the learnings and the experience of all this interacting with the healthcare system, I will never forget. But so back to genomes. So the early things, I think that we're really looking at in terms of like, how do we get this stuff into actually using it in the clinic and medicine? There's a couple avenues we're pursuing that only one of them I'll talk really in detail about. So the first is All of Us. So many of you have heard of the All of Us research program funded by the NIH. We are the largest genome center supporting that, and I'll give you a little bit of details in a couple slides, but that is a clinical implementation study as well as a research program. Another effort of sort of spearheading, genomics into clinical records is in the eMERGE study. So eMERGE is an NHGRI sponsored program where, and as well, the VA is working on this, as well as getting polygenic risk scores into the EHR of patients. And they're going to do this for tens of thousands of individuals who have signed up to be part of receiving a polygenic risk score for about 10 to 12 different conditions. And so this is ongoing, and I think really important early implementation work. Another thing, that we're working hard on is developing a rapid whole genome test for NICU situation. So we have the opportunity to learn from Stephen Kingsmore and his early examples, but we are thinking of now standing this up and sort of bringing scale to it. Some of these early rapid NICU tests have been like, 1 or 2 at a time, but what we do best, from what I tried to show you in the earlier slides, is scale. So we're trying to figure out how we would bring scale to this problem of extremely quick whole genome sequencing for NICU babies who need it the most. And then finally, a new opportunity has arisen recently to work with Quest. So Quest is a large commercial diagnostic lab, as you all know. And we've had great discussions with Quest, sort of getting to my early point, is that like, why do these panels? Like, why do test after test after test? Well, we could just have a genome as the ultimate test and keep going back to it for individuals. And so Quest has come to us and said, hey, can we work on implementing clinical whole genomes to replace our chromosomal microarrays as a place to start? They do tens of thousands of those every year, and a genome would be way cheaper and I think better information. So that's an area that we're exploring as well. But to say a few things about All of Us, because I think it's important for to really put out there when I'm in a room of genomicists, and especially young people who are really thinking about getting into the area. This is a really important resource, I think, for our whole field. So the goal of the program is that 1 million US individuals would be enrolled, would donate biospecimens, physical measures, their EHRs, survey information, and that this collection of individuals will be studied longitudinally over time. That's a really key feature of it, is that we would keep learning about what happens to the health of these individuals. Central to the program is diversity and really representing individuals that have been previously underrepresented in biomedical research. And the program is really doing an excellent job at that. I'll show you in a couple of slides. So where we stand right now is that about 700,000 people have put their hand up and said, I'd like to be involved. Over 500,000 have donated biospecimens. All of those individuals have had their genome sequenced so far. Not all of that has made its way into the database yet and to the individuals yet. We're working on that. But the latest data release is a couple 100,000 human genomes for individuals who have their EHR that are available through this researcher workbench that I point you to. And this is a really valuable and nicely set up tool. You can do things like cohort building. So search for who had diabetes, who didn't have diabetes at a certain age, and then pool information for those individuals, like lifestyle information if they provided it, their genomics, and so on. And we are looking to expand this beyond genomics. And so the idea is that we would also be layering on, in the fullness of time, plasma proteomics, RNA sequencing for some individuals, and even long reads. So we have a pilot of starting to sequence about 12,000 people with PacBio to further capture some of that important variation that Karen spoke about earlier. But a big part of this program, like the reason I pointed to it in the clinical implementation part, is that people are actually receiving information back from this program right now. So a really important part of the program is engaging individuals in what they can learn from their genome. And so people get the chance to opt in. It's not necessary to get it back, but to opt into getting health-related information back. So information about the ACMG, which is a collection of 73 genes, which have known variants that you might want to know about, right? That actually are pathogenic, that will impact, that most likely will impact your health, and pharmacogenomics. So everybody has pharmacogenomics variants that you might want to know about, right? For how you metabolize drugs. So right now we've returned health-related results to almost 100,000 people have received these tests back. About 3% of the people have received a positive test. There is something in your report you might want to know about and receive additional screening, talk to your doctor about. So that's pretty impactful. I mean, right now, this honestly in the lab seems like a very routine project for us. Hundreds of thousands of genomes are flying through the system, but to step back and think, wow, 3,000 people, in the past couple of years have learned something that they're actually going to do some, medically. Even people in our lab, not to reveal anything, but honestly, I was like, are they selecting us specifically for some extra level of analysis? But 3 people in the lab actually got a positive result on something that has resulted in some additional screening. So it's people I actually know, and it's really meaningful. And as I said earlier, really trying very hard to really think about diversity and increasing the representation. You all have seen the studies about GWAS is so woefully tilted towards individuals of European ancestry. So the program's doing an awful lot to increase that. So the data set right now is 45% diverse by race and ethnicity, and 77% underrepresented if you include things like geographic location, socioeconomic status, and so on. And so when you compare, across the different populations, the program's doing a heck of a lot, I think, to add to the diversity of individuals available for this type of research program. So that's my little plug for all of us. I'm very proud of the program and can't wait to see us get to the full million over time. So what else can we do, right? So it's always, earlier I had the chicken and egg, and I didn't do that earlier, but when you think about this this utility question, right? It always seems to me like, well, people would do more of it if it was interesting, but it's only going to be interesting if we do more of it, right? And have larger data sets and make more. So it does become this massive chicken and egg thing to me. And so what else can we do, aside from these early sort of implementation studies to do things like drive down costs, increase content, and really push forward in discovery? So just a couple of thoughts on that. One thing that we've had a lot of interest in at the Broad is this thing we developed called our blended genome and exome. So this is not rocket science. I'm going to put it out there, but it does solve a couple important problems. So what we did was combine sort of a higher coverage exome, so an exome that you can find rare variants, you can find variants in genes you might want to know about, so it's covered at about 60-fold. And then a low-pass genome that is sprinkled in there in the background. And with that low-pass genome that is around 3x coverage, you can call SNPs, you can do GWAS, you can do things like imputation studies. So the trick here, though, is that we're not just taking your sample and doing 2 things and giving you back 2 files. We actually take your samples, make 2 libraries, and combine them in a way that is balanced to give you sort of the best representation in both worlds. The reason people have been excited about this product is it's cheap. So we can do this assay for $99 a sample. And people want to use this for screening studies where they're not quite ready to invest in a genome, and where you might have previously picked to do an array study for like imputation or GWAS. But the problem there is that a lot of those arrays that you might pick have been built from individuals highly selected for European ancestry, right? So here you have the genome, and so you don't have to have that array that is really targeted to a very small part of the global population. So that's really how we're sort of marketing this. It's, again, $99 for research, uses way less sequencing. Oh, I had another point and I forgot. So anyways, we published the protocol. It's out there. We've had around 300,000 samples come into the lab for it so far, and I see another couple 100,000 ahead. So I think it's a big opportunity. Part of the publication involves an analysis of, how does this product do for exome calling versus like a regular exome? How does it do for things like imputation compared to arrays? And the answer is it does really well. And so you can kind of get a best of both worlds situation with this blended product. So beyond research projects, we are intending to move this into a clinical test as well. So this clinical test would allow you to end up with, again, this blended product that would give you a polygenic risk score in your report back, as well as any rare variants you might have in whatever selected set of genes you're interested in. And those two could come back for, again, this would be a screening test. I don't think this is a replacement for a diagnostic exome, but something that, certain cohorts that aren't, as I said earlier, aren't ready to commit to doing all the samples in their biobank at $500 each. But this is a way to dip your toe in, right, and see if this is something that you'd like to implement, in routine use. So anyways, that's the blended exome. What else? So how else can we think about cost and driving forward? And so I want to spend a little bit of time talking about this platform from Ultima Genomics. I'll say at the beginning, I have no financial, nor the Broad nor I have any financial interest in Ultima. We have just been collaborators now for a while, and I'll try to explain why. So no hard feelings to Illumina, right? Like everyone else in the room, I'm sure that we all have been, super big fans of Illumina and all they have done to drive forward technology over the years, and what a high quality platform that they have always produced. But, so I think we all recognize that we have been living relatively in a monopoly situation for many years now with Illumina. So here we've arrived now, so Illumina's latest instrument, this NovaSeq X Plus, is tremendous. The innovations they made from the NovaSeq 6000 to the X are amazing. This instrument will allow you to do something like 128 genomes in a single run. That's kind of crazy if you think about it. It's a 48-hour run. The instrument costs $1.2 million. They dropped the price, when this instrument came out, something that we've been waiting a long time to do. So if you see those, I know someone showed earlier, at least once, that how the cost of genome sequencing has declined. It did impressively for a long time. And if you look closely, between 2017 and about 2022, it was flat. And it was flat for a reason. It didn't have to go down because we had Illumina and there was no pressure, right? So finally, when the NovaSeq X Plus came out, they did reduce the price, their list price. So the list price was, a way we measure is dollars per gigabase, so dollars per billion bases. Before the X Plus, Illumina was around $6-ish. Now with the X Plus machine, it is $2 per gig as a list price if you use the high density flow cell, we should say. Great, so that's tremendous advance. But Ultima, came along. I can tell you more about that timeline in a couple of slides. The features here are that it's just different. I will say that the whole premise of the platform is very different in terms of the sequencing. The runs are faster and more often. That's one sort of highlight. And it's cheaper. So the premise here is that it is industrial scale sequencing only. So they're not intending to make sort of a benchtop or a NextSeq competitor. They want to be sort of in industrial scale labs. They launched officially a commercial launch in January at $1 per gigabase, so half of what Illumina charges. And importantly, they will tell you full-throatedly that's the start. They intend to get to 50 cents and then 10 cents a gigabase. So basically to the point where the sequencing is sort of an arbitrary thing. Like you don't even think about how much sequencing you need to do. You're just gonna do it. So how could that be true? We'll talk about a little. So what? Okay, so why, right? So why did we decide to look at this early on? And I think there's just great potential. There's lots of sequencing to do for lots of reasons. I've told you a bit about human genomes, but we'll get to the end about, and you've talked today a lot about things like counting applications, all the things you can do just by reading barcodes, just by reading short tags, right? And there's lots of reasons to do that in all of these sort of functional genomic studies. So when Ultima came along, it's like, wow, this could really sort of unlock that future to like really massive scale sequencing, way cheaper. Disruption's important. Like I said earlier, that flat part of the cost curve wasn't going to change, I think, unless there was some disruption. And the idea of it being industrial scale and these other sort of compelling features, I think really do suggest to me there is an actual lower cost of goods here. I mean, I don't know it. I've not like sat down with their accountants and really looked at what things cost them, but I believe the features of it, which I'll talk about here. So the biggest thing is that they have a different type of flow cell. So they don't use a flow cell. They use something called, they use this, they call it a wafer. So it looks like I'm going to date myself here, but we talked about birthdays earlier. It looks like a 45. Most of you don't know what that is, but it's like a record this big that used to go in a jukebox. In fact, we called this machine the jukebox at first because you stack up the 45s in the sequencer. So the thing about this wafer is that it's a semiconductor wafer, a silicon wafer that are plentiful because it's what the whole sort of computer industry uses, right, to build chips. So they're everywhere. They're super, super cheap. And it's unmodified. It's not a fancy sort of etched the little flow cells or etched the little sort of places for the alumina or oligos to go. It's completely unmodified. So that to me suggests it could be cheaper in reality. They have a entirely different chemistry. So you all know that Illumina is built on SBS, sequencing by synthesis, with these 4 colors, actually moving to 2 colors in the later instruments. This instrument does use fluorescent, mostly natural nucleotides with a non-terminating chemistry. So it's like 454, again, for those of you who might remember that back in the day, where you have runs of nucleotides and you read how many you have in a row. It's just a different chemistry completely. The fact that the nucleotides are mostly natural, not all modified, because some of them are modified with fluorescence, does make it a lot cheaper. And because they are selling the reagents in bulk, so no little kits, right? That also allows it to be a lot more inexpensive. And then there's less dead volume in the machine, all of that. And then another cool thing about it is that just the way they distribute the reagents saves money. They do this spin coating, so the reagents come down from a top. The thing is spinning like a record, and the reagents disperse across it. Kind of like if you've been to a county fair, like spin art, like that's kind of what it's doing. So it's all kind of interesting. So we got to know these guys and girls in 2018. Met them at ASHG through some connections we knew with some of their founders. We agreed to have an early prototype in 2019. We had this very secret time phase with them. We called it Project Jukebox. We never uttered their name because in this time frame, they were very worried about being sued by Illumina or being sued by whomever, right? Because they're a little company. So it was super secret, which kind of made it fun and feel like super stealthy, like the old days, like I can remember, I remember, I'm old enough, back in the days where, I walked into the Broad or Whitehead Genome Center and it was 45 ABI 377s, right, lined up in a row. And then we would get a new sequencing technology, then another one. We were always trying new things. And that has really changed over the past few years. But so it was kind of great to finally have the opportunity to work with something so very early. So we had different prototypes. We were able to complete whole genomes with them. The pandemic happened in here, as you all know. So we kind of got busy with that 38 million PCR, but still they stuck with it. We had prototypes. We did some productive things. So we've done cancer genomes with them. We've done the 1,000 genomes again with them. We're running Olink, which is this plasma proteomics that uses barcode sequencing. We're running that on this platform. And we've done single cell ancient DNAs, so various things. And they're now commercial, they're out there. So that's them. People are always like, oh, but the data is less good. We're really worried about the homopolymers, because it is true. This type of sequencing chemistry has very different error profile than Illumina, which we're all so used to after all these years. And their data, the first time we talked about it was not as good as it is today. So this is a big chart I can show you where we've been looking at sequencing these reference genome in a bottle samples and comparing sort of things like SNP performance, indel performance, and they've made a lot of improvements. So the first time we talked about it, I think at AGBT, maybe 2022, I don't know, the years all blend. They excluded like 2% of the genome to make their comparison, because they were trying to identify their high confidence part of the genome where they could actually say, this is where we're able to call variants. So that's now less than 1% that they have to exclude to make high confidence calls. They don't do great in the highly repetitive regions. So anything like a homopolymer over 12 or so, not going to do so well. They just won't call it, but you kind of know where those are, right? And for like, if you're sequencing tags or barcodes, you're not going to make your barcodes with 13 A's, right? So, you just have to, you design wisely if you want to use this platform. But but the SNP calling performance has gotten, and indel has gotten way better. So we're up at like 99.3% right now for the indel F1, looking at the genome in a bottle sample. So good improvement. We're not, to be clear, we're not replacing our Illumina sequencers with this platform right now. So we have, we're using this for targeted things. And the things we're using it for right now are identifying the use cases that really take the best advantage of that sort of scale and cost. So basically, like, what are the sequence greedy, the read greedy applications where you just want to do tons and tons of reads and you want to do them, as cheaply and quickly as possible. And so those things that I think about, not necessarily our human genome. Our human genomes are clinically validated, running on Illumina, everything's good. But there's these other things that we do, like when we're looking for really, really rare events, right? So you have this big signal to noise problem. And so you want to sequence very, very, very deeply to be able to tell a true variant from noise, right? So what are those situations? Liquid biopsy, where you have a rare event running around in a sea of normal DNA. Working with David Reich on some ancient DNA sequencing, kind of interesting. So he has like 10,000 samples that are like 20,000 years old or something like that. And with this ancient DNA sequence, you have this problem that there's a lot of contamination because they dug them up from archaeology. But you can imagine why it's hard, right? And why there's a lot of background. But we're able to do deep sequencing, and we're doing genomes with David in that category. All kinds of counting applications. I'll talk to you about a couple of them in like 5 minutes total. And then anything where you're monitoring. So I always think about these applications like liquid biopsy or MRD, where you want to be testing a patient over and over again to see if things are changing, if you're looking for minimal residual disease. And so something like that, where you're going to be able to want to do like some amount of whole genome sequencing like every 3 months on an individual, you might want to do this kind of very cheap, fast sequencing in the end. See, I talked a lot. What are the other things that we want to do? Like, what are the other technologies that kind of are away from the clinical and the, most of what we do? And I think getting to really this question of going from genes to gene expression to how cells function and then to health and disease. So obviously a big, focus is single cell. So I couldn't decide if I wanted to show you like the fun, how you think of single cell differently from other things. Because I just love this analogy, So most RNA sequencing is bulk, where it's all mushed together. Single cells are like you can taste the individual fruit. And then spatial, which I don't have time to get to, but I would love to talk about it, is when you have this beautiful arrangement of the individual fruit and who's next to each other. The other way to look at it is you can do bulk and it's all mushed together in a tube, or you can measure sort of gene expression in single cells and find out why things might be different, or a cell that might have been perturbed differently behaves differently in sort of RNA sequencing land or whatever it is. So this is all really key, I think, to following up a lot of, from the association studies, we have targets, we have genes. What are the function? What are the important variants? What are the pathways, right? And so, single cell sequencing is really what I think enables us to get to this resolution. But at even higher scale, so, the initial, I would say, single cell experiments like the atlasing things, again, this field has come a tremendous. I was looking for a plot. I couldn't find one of like how much single cell has advanced in the past few years. I mean, it was, I think 2009 and 10 were some of the earliest, where I remember Aviv got a paper published with 18 cells, right, in 2009 or something, which is amazing. And so that's not that long ago. But if we really think about how this might scale, and it's really with some of these very large, I would say, systematic efforts that might want to profile many cell types with many conditions and do it a couple at different time points or so. Like when you get to those kind of, I think, functional applications of single cell, you're really talking about needing millions or billions of cells that you can analyze. And so how are we going to, push on that? And so, those experiments we can think of in the following way. So if you want to understand how genes affect, might change it with different expression, there's lots of perturbations you might do with these CRISPR screens, but you've heard about this today already. I won't elaborate. You just want to measure expression. And so those are Perturb-seq. We also are talking about this sort of neat assay, which is understanding the functional effects by optical pooled screening, which is not traditional sequencing. It's actually in situ sequencing. I'm probably not going to have time for it, but it's another example of how you might apply that sort of perturbation, a genotype matches some phenotype at massive scale. So the thing that I'm really excited about in this space right now is again the scale. If you think about how this field has progressed, you sort of started with the plate-based approaches where you would have tens to hundreds of cells. Moving to some of these microfluidic emulsion approaches, you have 10x, you have sort of other sort of nanowell approaches where you get to tens of thousands. But then there's these newer things like the split pool barcoding or templified emulsions, which I think really can catapult you now to like routinely million or billion cell experiments. And just so the reason that's important, obviously, the different single cell approaches is sequencing can get as cheap as we want it to get, maybe, but it was always going to cost a lot to prep the cells. Well, so what, right? It's still going to limit how much I can do. So that's really led to our interest. Again, I have no financial interest in this company, Fluent, but the Broad has no interest. But we do have a nice collaboration with them. And so this is a small company right now that there's a paper they published not too long ago on this method called PIP-seq, which stands for, I always have to write it down, particle templated instant partitioning. That's easy to remember, right? So basically, it's just a strategy to barcode your mRNA in the cells so that ultimately the material can be pulled and sequenced. So it's a very simple processing that they actually use here. There's no specialized equipment. You need a vortexer, right? And you need some beads that you buy from them, and then you need your cells. So this is kind of really cool because I think it's going to create access. It's scalable because the amount of cells you can capture scales with the container volume, essentially, and the size of the bead, not like how many cells you can pass through your microfluidic device, right? So the fact that this can be done so much in parallel and with simple instrumentation really makes me think you can get a lot of cells. Another important thing about this one is that once these cells are emulsified, you have the cells, you have the oil, you have the beads, you can do that somewhere and then put them in the freezer, put them aside, and then send them off for processing later. So that's kind of awesome compared to, I think, the 10x workflow, where it is hard to really store anything because you kind of have to get it straight from when you encapsulated to putting onto the 10x device. So this could really, again, open up the usage of this kind of profiling and let you maybe even think about doing this sort of more like in the field or in the hospital or in different places. And the fact that it can be tube or plate based, so you can do this on a plate, really, I think, speaks to the ability to potentially automate it, which they've not done yet. Again, this is a small company, but something I think we'd like to work with them on. So anyways, check out the paper. I think it's really interesting. So we've done, this PIP-seq or Fluent approach combined with the Ultima sequencing. Aziz Al-Kavaji in our lab worked with folks from Vijay Sankaran's lab at the Broad and Dana-Farber. We started with a CRISPR library, so about 1,200 guides transduced into these RPE cells. You add your Cas9 for the nucleofection, perform the Fluent analysis and sequence on Ultima. They had really nicely performing Perturb-seq data here based on the number of cells we could identify, which was 275,000 cells in this experiment, with 26,000 transcripts per cell and 5,000 or 6,000 genes detected per cell. Other things you look at are the amount of guides that were detectable and how many guides in the library were detected. And again, they had good measures there. So we're encouraged that I think this very simple approach can be matched to very cheap sequencing to hopefully allow really high scale. It won't get through that part of the optical pooled screening. I think is interesting. I encourage you to look at some of that work out of Paul Blaney's lab. I just wanted to end with a exciting, and I have no data here, I'm just something we'd like to do with some of this very high scale Perturb-seq and high scale optical pooled screening. And it's a systematic approach at uncovering pathways and targets for rare disease. And this is a program that has been dreamt up by my colleague at the Broad, Anna Greca, and we recently put together an ARPA-H proposal around this. So she calls this Project Radical. Radical stands for Rare Disease Cures Accelerator. I'll let you do the little acronym in there. And the idea here is that we're going to take the 8,000 known rare disease genes. We're going to take 3 known pathogenic variants in each of those 8,000 genes. So that's 24,000 variants that we're going to create CRISPR libraries for. We're going to have 10 cell lines. And then we're going to do a big Perturb-seq and OPS experiments, measure RNA sequencing, measure cell morphology, cell phenotypes matched to whatever CRISPR guide got into them, with the idea of finding signature pathways that you might be able to lump multiple rare disease genes into, right? And once you find these pathways then that are sort of, she calls them nodal pathways that might connect multiple types of diseases, you might then move those into cell line models where you can perform genome-wide CRISPR-I and try to find genes that sort of rescue that phenotype as potential targets. And again, this is sort of the idea being that these are systematic approaches. You don't have to go. The idea is not to do the 24,000 variants one at a time, but to find approaches where you can do them sort of in a pooled fashion and measure them in parallel. And so there's an awful lot of computation that goes into this. I cannot even come close to explaining all of the things that are going to happen on that side. But this is the type of, I think, multimodal and sort of rich, deep profiling experimentation that we want to be able to get to, with these improvements in sequencing and single cell technology. So I'm going to wrap up. There's more future than past here in genomics. There's a lot more to do with all of this. So I'm envious of all of you that are entering at this really incredible, exciting time for technology. I mean, I'm someone who poured agarose gels in the lab, right, and studied one marker at a time. So I'm jealous of those who are never going to go through that and have these amazing tools available. I'm always going to be interested in seeing us drive towards more and more utility and more equitable access to genomic technologies, particularly when it comes to clinical adoption. We got to be really careful about data sharing. I didn't get into any of that, but I see something like this about, so there's been this executive order about protecting our data, right, from countries that we might not trust. And one of the things that the first type of data that's listed is genomics. So I don't know, I'm worried about that. And we should be really, really careful. And we should make sure that the data that we're generating and putting out there is very well protected and that the public understands it's very well protected. And we should really mean that when we say it. So something I've been thinking a lot about, because I don't want that to be something that stops studies like all of us from happening. We can't take our foot off the pedal when it comes to cost. People hear me, Illumina is so sick of me talking about cheaper sequencing, you can be sure. But I'm excited about it because I think this is what moves the needle forward and a lot of uncovering a lot more discovery that we need to. And these technology development programs are why we get there, with so many of these new technologies. And so, yeah, huge fan and thank you for all you do. And I have a great team, a large team of people in the lab. But yeah, that's us. Check us out. We have websites and everything. But thank you.
I will put in a plug for NHGRI by saying that Ultima had an SBIR grant.
Oh, that's right. I knew that.
In their career. Now they had a little investment after that to bring that to market, but that was one of the things that really helped them get started.
Now that you say that, Mark, I remember that. So yes, that's right. I should have said that.
Quick question for Stacey.
Thanks for the really interesting talk. Kind of building off one of your last slides with the sharing of genomic data as a public security. It's my understanding that currently insurance companies can't deny you healthcare coverage based on these results, but I believe you can be denied disability, long-term, and life insurance. So are there protections for the All of Us program? Or, I mean, obviously it would be so great for clinical settings to do these genomes, but is there protection? Do we know of any like laws that are being proposed to protect patients?
Yeah, I mean, that's something that that the program does talk quite a bit about. I mean, we do try to make it clear to the patients that, this data is sort of owned by the, Vanderbilt, that the Vanderbilt's technically who owns the database where the data sits, that, it's not able to be accessed, by insurers, for example. It's not out there. But I think there is concern. I mean, there's obvious concern. I mean, we ourselves can't pass any of those laws or lobby for those laws to be passed, but I certainly think that's a concern, that it doesn't extend beyond healthcare, because, that's going to inhibit people from finding these things out about themselves. It ultimately should save the healthcare.
Right, it could be preventative medicine.
That could be preventive, right? And so, no, I think we all need to do a lot more about that, but I don't have a good answer for you other than, yes, it does. Beyond just the health insurance, I think that it's not a great situation for us to live in right now.